Bismillahirrahmanirrahim. It's me, Muhammad Hamas from BS Chemistry Department. Our topic is formaldehyde. Formaldehyde is the simplest aldehyde and its 40% aqueous solution is known as formalin. It is a colorless poisonous gas with a strong smell that causes a severe damage to our central nervous system and immune system. It is generally used in making of many resins, polymers, wood products, and protein cross-linkers for tissue fixation. The thermal and chemical decomposition of construction materials such as particle board and urea formaldehyde form insulation-heavy release Fa in the atmosphere. Fa is a well known for its toxicity nature. In humans, formaldehyde transforms into formalic acid, giving rise to breathing difficulties, coma, or even death. Recently, International Agency for Research on Cancer has announced formaldehyde as one of the main group of carcinogenic organic compounds in the world. The World Health Organization declared the limit of exposure to the formaldehyde as 80 parts per billion as a standard for 30 minutes. Occupational Safety and Health Administration fixed standard value for permissible exposure limit at 750 ppb, while maximum exposure value for intermediate dangerous for health is considered as 20 parts per million. Environmental protection agencies had determined the maximum reference dosage of Fa is 0.15 to 0.2 mg per kg body weight per day. In the brain of healthy individuals, the concentration of Fa is in 0.2 to 0.4 mm range. In body tissues, Fa reacts with biomolecules like amino acid, protein, nucleic acids, nucleotides, and produce unstable assemblies which transport Fa to distant tissues from the respiratory tract. Consumed Fa cause damage in liver and kidney, which leads to jaundice, acidosis, or individual in central nervous system leading to depression or even heart failure. Due to the fact it is highly dangerous for the mankind and nature, it has become very important for detection at low concentration level. Nowadays, many techniques used to detect its efficiency. The first is detection of Fa using small molecules based sensors. Fluorescent imaging has become the crucial part of recent scientific discoveries due to its ability to observe changes inside the cells and tissues. Consequently, scientists have used this method for fluorescent detection for rapid and accurate detection of Fa. According to the recent research, in the most cases of formaldehyde detecting fluorescent probes, the amino groups of the probe and the carbonyl group of HCHO generally react to detect formaldehyde via colorimetric changes. The detection-based regions are generally categorized into two classes: formamine type of intermediate forms from the reaction between Fa and primary amine of probes. This consists of three types: 2-AZA coop rearrangement, subcontinent of beta elimination from the probe, aromatic hydrazine amino formation via reaction of Fa. Using these reactions by their functional modification, Fa sensing mechanism can be attended by a different process. The first is 2-AZA coop rearrangement based fluorescent probe. AZA rearrangement reaction is currently used as a successful method to detect one bearing a fluorescent probe react with formaldehyde. 2-AZA sigma-tropic rearrangement reaction take place. As in figure 2a, amine groups. Amine groups of probe react with Fa and iminium intermediate is formed, which then undergoes sigma-tropic rearrangement reaction, which gives rise to the fluorescent product after hydrolysis. The second step is aromatic amine-based fluorescent probe. Its general formula is RCH double bond NR are made by a simple condensation reaction between amine and aldehyde. These reactions are precisely used for the generating C and bonds in the organic chemistry with a very high yield. It is used as one of the most suitable and useful ligaments for the detection of metal ion and some organic material due to their strong coordinate capability. And the third and the third one is hydrazine-based fluorescent probe. In past few years, several fluorescent probes with the hydrazine moiety were developed for the selective detection of Fa. Hydrazine moiety with an amino group has a very strong nucleophilic which shows very high reactivity for Fa. Upon reaction with Fa, it forms active methylene which reacts with PET process from the hydrazine. The figure shows Fa detection which has a very weak fluorescent at 467 due to PET process. From amine to the chlorine fluorine, however, after adding Fa, the fluorescent intensity increased by 5.5-fold due to the elimination of PET effect. The probe shows high selectivity in the process of several other organic compounds such as acetone. Other aldehyde form acid and acetic acid. The detection limit of probe was determined as 900 nm. The probe was able to successfully detect Fa in several food samples. Nanofibers were fabricated by blending the probe with the followed by the electrospinning. Moreover, upon immersion of the nanofibers into water combined Fa was noticed. Another probe was also reported by Yuan and co-workers for the extreme selective and selective detection of Fa. The fourth is Fa detection by the amino formation. An amino group is a type of organic compound having two amine groups attached to the same carbon atom. It has a general chemical formula CnR2nR2. When Fa reacted with its defined moiety of a condensation reaction took place, an amino intermediate was formed. The intermediate consequently opens the intramolecular, thus forming a strong fluorescent rings. A rhodamine-based fluorescent probe was synthesized by Li at Al. The detection of Fa using polymeric probes. Most of small molecular probes are less soluble in aqueous medium, which limits their biological applications. There is a need of polymeric sensors with good water solubility, high biocompatibility, low detection limit, and rapid sensing ability. A lot of organic polymeric probes have been used in the form of coating material. In 2010, Finch and co-workers reported the first sample jet sensitive yet sensitive upon the interaction of the Fa amine group present in the polymeric probe with Fa. An alternation of pH occurred due to which the indicators showed the change, whereas for the aldehyde, almost negligible changes were observed after the exposure of 5 minutes. This was the first report where Fa was detected by a naked eye. In recent years, water-soluble supermolecules in past few years, sovereign probes with the hydrazine moiety were developed for the selective detection of Ma. This table shows the amino acid of DTA resulted super self-assembly in water through the hydrogen bonding interaction. When several aldehydes were added to DTA, only in the case of Fa, the state of DTA changed into the colorless solution and the light blue fluorescent was observed under the UV lights. Moreover, The morphological changes for the fibers instruction was noticed after the Fa addition as a sample shown in the figures. Fa and the detection limit of a sensor was evaluated to be the around of 0.05 ppm and emission intensity variation was detected with only 1 ppm of Fa, which further confirmed its sensitivity. Moreover, they successfully applied the sensor to detect HCHO in the tap water and three other commercially available food samples as chicken, bream, and pork. The Arista and co-workers for the detection of Fa. The device was extremely selective for Fa and could sense Fa vapors at the precisely small amounts and comparatively extensive range of concentration of 0.005 to 5.0 ppm. Direct interaction of graphene surface of Fa vapor was the main reason for the sensor response. The polymeric material acted as a range of concentration. The polymeric material acted as an adhesive material when cast on the film. PMMA with a more hydrophobic nature extremely deters H2O from the polymer surface and therefore from the graphene sheet surface which cuts the sensor response to the water molecules. However, Fa has the additional hydrophobic interaction capability compared to the water molecules can successfully pass over the polymer's repulsion wall and the result in the signal in the graphene sheet. The limit of detection of its material was calculated to be 10 ppb. Sayed and co-workers used a thorough responsive functionized silica nanoparticle probe 90 for the selective chlorinic sensing of Fa. In the probe, the thiol groups interacted with the sequential dye detection dye observed only when both thiol and polymer groups were attached to the silica. The bulky polymer group CRE attached to the high polar around the thiol as shown in figure. Due to the bulkiness of aldehyde, no changes was observed. The LOD of Fa was calculated to be 36 ppb, able to. Now we have discussed the conclusion and future perspective. The synthetic review of the development and evaluation of the different types of small molecular and polymeric probes for the sensitive as well as selective detection of Fa, with a special emphasis on the advantages of polymeric probes, is present here. Through this comprehensive study, we concluded that different types of fluorescent sensors were designed and developed by using the mechanism of ACA-Copley arrangement of Fa-amine condensation reaction for extremely selective sensing. Mounting evidence indicates that among different methods has emerged as a significant platform for detecting Fa as a visible as a vi- as a visible extremely selective sensing. Mounting evidence indicates that different types of techniques, the reaction-based Fa as a visible color changes appear after selective addition of Fa, which in turn is important for real-world application. Furthermore, various research groups have developed different types of small molecular probes and effectively use them as a fluorescent imaging agent for the detection of oxygenous as well as endogenous formaldehyde in various living animals. Apart from small molecular probes, researchers have explored the potential and advantage of using polymeric probes to overcome the problem of poor adequate solubility. High detection limit and longer exposure time suffered small molecule probes. Despite having despite having a promising application, some challenges still exist for further revolution this field. A major bottleneck for developing a polymeric probe is their synthetic procedure. It is difficult to synthesize large-scale polymer due to long synthetic pathway. Investigations are underway to overcome this problem to synthesize a polymeric material in industrial scales to detect the mitigate and harmful effect of FA in our daily life. Furthermore, after FA addition, there is always the problem of biodetergent. In this regard, amino acid terminated polymers are evolving as a potential solution. Through developing novel polymeric material for selective FA detection have gained significant attention of research in recent years. Only a few reports are available where polymeric materials have been successfully used in biostudies. We believe that a lack of understanding of reaction mechanism between polymeric probes and FA restricts their full potential. In addition to this, in most of literature reports, the synthetic sensing systems are single use only. This limits their application for environmental monitoring, where continuous for repeated monitoring play an important role. Therefore, a thorough investigation of two in vivo behavior is most requirement for the clinical application as suitable agent. Yet, the finding of novel polymeric material have groundbreaking push to the quest for new operative treatments for the chloroquine. We are hopeful that in-depth discussion of FS sensing pathway and recent development of polymeric probes have encouraged the scientific community to overcome and find the permanent solution of FA. However, further studies were needed for developing the probable particles solution material have successfully in numerous discussions. Thank you.